I hope all of you are doing well. I'm back with our next class. In this class, we are going to discuss a topic which is very important, and that is megasporogenesis. Alright? How the embryo sac or female gametophyte will be developed. So let us start. In the last class, we have discussed about various types of orientation of ovule, right? There are various 6 types of ovules that we have discussed: anatropous, orthotropous, hemitropous, etc. So the most common being anatropous, right? So say this is the ovule where a female gametophyte will develop. How it develops, that is where we are going to discuss in today's class. See, in the ovule, this portion is called the nucellus, isn't it? Right? It's a mass of parenchyma tissue. Now, these being the micropyle, right? And this being the salazar. These points you have to remember, because I'll be making various reference with respect to these points throughout the discussion. Alright? So now, during the female gametophyte formation, one of the cell of the nucellus, which is nearby to the micropyle, it will start to differentiate. And while it differentiates, suppose I am making the cell here. This is the cell of the nucellus nearby to the micropyle, and it has differentiated. While it is differentiating, it is going to be forming a archisporial cell. Archisporial cell. This archisporial cell, of course, is twice M or diploid. Alright? The archisporial cell further will differentiate, or rather, it will undergo periclinal division, giving rise to 2 cells. This is towards micropyle, this is towards salazar. Okay? Towards micropyle, it is going to form primary parietal cell, and towards salazar, it's going to form primary sporogenous cell, just like we have discussed in microsporogenesis. Alright? Now see, the primary parietal cell do not have any function here. It would just remain or degenerate. So basically all the development is going to happen in primary sporogenous cell. So suppose this is the primary sporogenous cell, which will now convert, or it will be directly going to develop into the megaspore mother cell. Like in microsporogenesis, you have learned about a spatial cell that will give rise to pollen grain is pollen mother cell, isn't it? The pollen mother cell will undergo meiosis and give rise to pollen grains. Here, the primary sporogenous cell will directly behave as megaspore mother cell. Now, this megaspore mother cell will undergo meiosis and then give rise to the megaspores. Alright? And the megaspore, once formed, will eventually give rise to the whole embryo sac or female gametophyte. Alright? So let's see how it would happen. Now see here. Suppose this is the megaspore mother cell. Alright? Megaspore mother cell is twice M diploid. It will undergo meiosis first. As it undergoes meiosis, it will give rise to 4 haploid megaspores. 4 haploid megaspores. Because the 4 haploid megaspores are going to be linearly arranged, alright? They are going to be linearly arranged like this. Suppose Suppose this is the megaspore mother cell and it gives rise to 4 haploid megaspores which are going to be linearly arranged, and we call them megaspore tetrad. Okay, megaspore tetrad. Alright, now this is the micropylar end, this is the salazar end. Alright, now mostly what will happen, you see, 3 of the megaspores that are towards the micropylar, they will degenerate. Only one of the megaspore that is extremely towards salazar, that will survive. That means these 3 will degenerate. Only the one that was towards the salazar, that has survived. Understood now? So now, this is the only cell or the megaspore that has survived. First of all, this cell will undergo expansion. It will expand. Micropylar end, salazar end. It will expand. Why? Due to a vacuole formation. But by the time a centrally located vacuole is being formed so that the cell is expanding, do remember this haploid nuclei will undergo one round of mitosis also. One mitosis will occur while the vacuole is formed, and while the mitosis will be happening, 2 nuclei will be formed, isn't it? So those 2 nuclei, as soon as they are formed, they are going to be pulled towards opposite pole. One will go towards salazar end, one will remain towards micropylar end. Why? Because in between them the vacuole is being formed, and that's why the cell is expanding also and separating the 2 nuclei towards opposite poles. Understood? So one round of mitosis will occur in the nuclei of the megaspore that has survived, and as soon as the mitosis happens, 2 nuclei will be formed like this, and these 2 nuclei will be then pushed towards opposite pole. Why? Because in between them a vacuole will develop. Understood this? Now, being at opposite poles, another round of another two mitosis will take place. Alright. So total how many mitosis happening? 1 here, 2 here. Alright. Another 2 mitosis if takes place, then what will happen? So this is the one nuclei, this is another one. This is salazar and micropylar. Right? तो एक mitosis होगा, तो this is what will be the condition. Here I am telling 2 mitosis will happen. That means they again will undergo one round of karyokinesis. So after 3 rounds of mitosis happening in a successive manner, we'll get 4 nuclei each. Okay, at opposite poles. Towards salazar end also there will be 4 nuclei, and towards micropylar end also there will be 4 nuclei. Alright. Now eventually, one nuclei out of the 4, 1 nuclei from salazar end will come down at the middle, at the center, and here also from the micropylar end also 1 nuclei will move down at the center. So from here out of the 4, 1 will go to the center, and here out of the 4, 1 will go to the center. Okay? So now finally what will happen? Or let me show it here itself. See, these 3 nuclei will get surrounded by thin layer of cytoplasm. These will also get surrounded by thin layer of cytoplasm. Alright. So towards salazar end, the 3 cells that will be formed, they are going to be called as antipodals. Alright? Towards micropylar end, also 3 cells are formed. Alright? They are going to be actually like this. They will share the wall like this. Okay? And hence, out of these 3 cells, this middle one is called the egg, the female gamete, and these 2 on the sides of the egg are called synergids. Synergids. Egg and synergid together makes up the egg apparatus. Alright? And these 2 nuclei, they won't be getting surrounded by any thin peripheral cytoplasm as such. They will remain in the nuclear state and we call it polar nuclei. Polar nuclei. So this is the most or the well-developed embryo sac or female gametophyte that has been developed from the megaspore mother cell. So a question can be asked that how many of mitosis and meiosis in the megaspore mother cell will give rise to a well-developed embryo sac? Your answer is 1 meiosis. 1 meiosis and 1 plus 2, here 3 mitosis will give rise to a well-developed embryo sac. Now in the well-developed embryo sac, you see here what is happening. Towards salazal end, we have 3 cells called antipodals. Towards the micropylar end, we have 3 cells. In between, in the middle, we have egg. On the 2 sides, we have synergids. And in between, we have 2 polar nuclei. All the cells present inside the embryo sac are haploid. So this is a female gametophyte where everything or every cell present is going to be a haploid. Alright? Now, a very, very important thing that you should know is this. Suppose this is the embryo sac, this is the salazal end, this is the micropylar end, this is the egg apparatus. These are the antipodals. And these are the polar nuclei, right? This well-developed embryo sac is going to be known as 7-celled, 8-nucleated embryo sac. 7-celled, 8-nucleated embryo sac. How? There are 8 nucleus present here. 1, 2, 3, 4, 5, 6, 7, 8. Simple. Where are the 7 cells? 1, 2, 3, 4, 5, 6. 6 cells because they have nucleus also and they have cytoplasm also. So we can consider them to be cell, isn't it? So 1, 2, 3 cell. Here 1, 2, 3. 6 cells. Where is the 7th cell? This is the 7th cell. This middle part. This is called the central cell, which makes up the 7th cell of the embryo sac. That's why we call it 7-celled, 8-nuclear embryo sac. Alright? So in this entire embryo sac, out of the 7 cells, these 6 are going to be haploid. But the central cell, it is not haploid. It is binucleated. Or else, sometimes what happens? The 2 polar nuclei might fuse together before fertilization. It might fuse together, forming a diploid secondary nucleus. Okay? So, if these 2 nuclei will fuse together, forming a diploid secondary nucleus, in that case, the central cell becomes diploid. Understood? So, in the embryo sac, these 3 cells and these 3 cells, antipodals and egg apparatus, are going to be haploid, but the central cell is not going to be haploid. It's going to be either binucleated or else it is going to be diploid. Alright? So this process, how the megaspore mother cell undergoes meiosis is going to be referred as megasporogenesis. Is that clear, all of you? So this is for your today's class. In the next class, we are going to discuss about pollination. Thank you.